Hi, I'm Kevin Liu, a faculty member at Michigan State University. I'd like to begin by saying thanks to the session organizers and to the conference committee for putting this fantastic conference together and for giving me the chance to talk to you all about some of our lab's recent research. I'll be talking about a new resampling method and a new phylogenetic support estimation method, which we're referring to as RAWR, short for random walk resampling. I'd also like to acknowledge the lab members who have really pushed forward this work. These include my current students, Wei, Julian, and Ahmad, as well as my former students, Hussein and Jack. A quick brief aside, if you're on the lookout for a phenomenal researcher, my student Wei is actually going to be graduating this fall. Feel free to reach out to me if you've got some suitable opportunities that you'd like to discuss. And so my talk today is really going to focus on a really important task in systematic biology and evolutionary biology, which is trying to reconstruct phylogenies and then estimate support. And so this really pertains to trying to look at rigor and reproducibility. To what extent can you actually reproduce the phylogenetic estimates that you get on the data that you have? And really a de facto standard for trying to assess this is an approach which is referred to as the phylogenetic bootstrap method, proposed back in 1985 by Felsenstein. At this point, this is such a widely used method. It's accrued so many citations. It is now the 41st most cited paper in history. In brief, the way the phylogenetic bootstrap method works is as follows. From your input, which consists of an annotation, multiple sequence alignment, and phylogeny, what you do is the following. You resample the columns of your input MSA using the standard bootstrap method, which is simply just take your columns and just uniformly at random resample them with replacement. And so this requires a key assumption, that being that your input columns in your data are basically independent and identically distributed. And so long as you can make that assumption and perform your resampling, then what you do is on each resampled data set, you re-estimate your phylogeny using your phylogenetic estimation method. And then finally, you repeat this enough. And then finally, what you do is you compare the re-estimated trees versus your annotation tree and simply report the support being for each bipartition in your annotation phylogeny. You calculate the proportion of the re-estimated trees that actually display that bipartition. And so, as I mentioned, the application of Bootstrap in this context really requires the assumption that your data are IID. But there are a number of factors why this is basically an oversimplification when it comes to biomolecular sequence data. These include things like biomolecular structure and function, recombination, as well as sequence insertion and deletion processes, as well as others. And so in each of these cases, the assumption that your data are IID is really limiting. And so the loss of the sequential nature of your data is really kind of a problem when it comes to Bootstrap resampling. And so to deal with this, there have been a number of alternative resampling methods that have been proposed. And one such method that I'll mention is a method proposed by Landon and RAWR that's referred to as the heads or tails method. It's rather straightforward. They only looked at this in the context of multiple sequence alignment, and here's how they used it. So from the input being a set of unaligned sequences now, you can apply an MSA method, get your MSA estimate. That's what's shown on the left. But what they reasoned is, if you instead mirror your sequences, rather than reading them left to right in the traditional way, instead, if you read them from right to left, then pass that as input to your MSA method, you would hope to get the same MSA, the equivalently same MSA as output, but you actually might not. And to the extent that you don't, this basically gives you a reliability check. And so this particular approach, it has, of course, a nice advantage in that it's simple, but some disadvantages being that you actually only get two estimates to compare. And furthermore, this is just really proposed and studied in the context of multiple sequence alignment estimation. So to address this, Landon and RAWR and some of their co-authors, they basically extended this with special purpose parametric approaches to deal with some of these limitations. But really, what we wanted is to have kind of a similar, simple, general purpose alternative, which could be used for a lot of different applications. And so to try to make progress towards that goal, we actually had a few different insights on the back of that contribution that Landon and RAWR put forward. First off, what we realized is that we could, instead of resampling from the starter ends of the sequences, we can simply do so from the interior. So for example, in this particular illustrated example, pretend for a second, for the sake of discussion, that you have a set of aligned data. And now here you're sampling from the interior of the sequences. picking a direction just as in heads or tails. Here in this example, you're reading from right to left in the reverse direction. And just like in heads or tails, you keep going until the end, okay? And so I'm showing here at the bottom the unaligned data that results from this particular first insight. Now, if you can read from one end of the sequences to the other, or vice versa, you can combine this in the other direction to basically continue our random walk in the opposite direction if you hit the ends of the sequences. This is simply just essentially reversing your reading if you get to the ends of things, or reflection in that case. And so the alignment problem is still going to be meaningful for globally alignable sequence inputs if you do this second insight as well. Finally, we had the insight that if you can reverse the redirection at the ends of the sequences, then you can do so also at the interior of the sequences as well. So here I'm showing you such a reversal here inside of the sequences, not just at the start or the ends. And you can do so with some probability, let's call it gamma. And the resulting procedure basically takes the form of a random walk on the sequences. We refer to this procedure as RAWR, short for random walk resampling. And so in essence, what we can see RAWR to be is basically the synthesis and kind of an extension of both Bootstrap resampling and the mirrored inputs idea of Landon and RAWR. And so the way that we actually do this random walk procedure, it is parameterized by a few different things, including the reversal probability and exactly what convergence criteria that you use. But in general, this is a pretty simple procedure that is very general purpose and flexible. So these are some of the properties that we're looking out for. So you could use them for a lot of different applications, but in this particular study, we focused on just the application of phylogenetic support estimation. And here's the pipeline, the results when you use RAWR resampling in this way. So to begin, you've got your set of unaligned sequences from which you compute your annotation, MSA, and tree using an MSA estimation method and a tree estimation method on that MSA. And then from the annotation MSA, you basically conduct this random walk procedure, just like how I discussed, to get your replicate set of unaligned sequences. And on that, you re-estimate your MSA and tree as shown. And just like with all of the other traditional resampling methods, you have to repeat this enough to get enough resampled data sets on which you can perform your re-estimations, resulting in a set of re-estimated trees, which you can then compare to your annotation and MSA and tree in the traditional way. That basically being just computing support for each bipartition in an annotation tree, what proportion of re-estimated trees actually display that bipartition. That's going to be the output here. And so what we then set out to do was to study the performance of this RAWR method as applied to phylogenetic support estimation using both simulated and empirical data. So the simulations were performed using indelible rows under a range of model conditions. These included a range of dataset sizes ranging from 10, 50, or 100 taxa, sequence evolution under a finite sites model of nucleotide substitution, along with either a medium or long gap length indel model. Furthermore, in addition to the simulated benchmarks, we actually also had an empirical benchmarks, which we had taken from the comparative RNA website database. These are really nice because it's a phenomenal resource because it comes with these intensively curated reference alignments from which we computed reference trees. And so using these benchmarking data, then we set out to compare the performance of not only RAWR, but other existing methods, including the Bootstrap, as well as a new method proposed by Lemoine et al., which is referred to as the transfer Bootstrap expectation method. For estimation of MSAs and trees, we used either MAFT or ClusLW for the former and maximum likelihood estimation using Raxamol for the latter. The different methods were compared based off of their type 1 and type 2 error, measured using precision recall curves, an area there under precision recall curves. And we also assessed runtime and peak main memory usage as well. Here are the results from our simulation study comparisons. I'm just going to show you results on the 10 taxon model conditions first. These model conditions are named 10.A, 10.B through 10.E in order of increasing evolutionary divergence, generally increasing divergence. Here I'm showing you a comparison based off of area under precision recall curve for just two methods, Bootstrap and RAWR, where I'm showing the best area under curve here in bold, as well as assessments of statistical significance based off of a one-tailed pairwise t-test with multiple test correction. Got the corrected Q value shown here in the last column. And so what we can see is that for these 10 taxon model conditions, for all of these cases, RAWR is giving significant and increasingly large improvements in area under curve, especially as these model conditions get more divergent, and therefore the alignment and tree estimation problems get more difficult. That's on the 10 taxon model conditions. Here's what we see on the 50 taxon and 100 taxon model conditions. So the layout's basically the same. In the situation, the comparison is roughly the same too. What we see is that on the least divergent model conditions, you see that more or less most of these two different methods give similar area under curve results, but as divergence increases and estimation gets somewhat more difficult, then RAWR returns significant and increasingly large improvements in area under precision recall curve. And so its performance advantages, at least in terms of type 1 and type 2 error, tends to increase in that case. That was for the simulated benchmarking data. Here are the results on the 6 empirical benchmarks, which consists of intronic ribosomal DNA data sets from the CRW database. And in each of these cases, the area under precision recall curve returned by RAWR was an improvement upon Bootstrap for these 6 different benchmarks as well. In addition to the performance study, we also set out to reanalyze previously published study from Lamachan et al.'s 2015 publication shown here, where they looked at a really charismatic clade of species referred to as Darwin's finches. So this is interesting for a whole lot of different reasons, but for us, we're really interested in a particular question, that of phylogenetic uncertainty in their original analysis. And so what we set out to do was to use RAWR to reanalyze whole genome sequence data that they had posted alongside their study. Here are the samples from which the sequences were drawn. Here are a plot of the different taxa under study. And they're basically from the Galapagos Island and as well as an island just outside. Briefly, I'll just mention that the different species, some of them occur across a bunch of different islands within this region, and others not. Furthermore, I'll point out that the coloring is actually meaningful, green, red, blue, etc., where the coloring basically has this correspondence. I'll really just draw your attention to one in particular. The species and or taxa, which are colored green, are those corresponding to the tree finch group. And so these tree finches here are identified along this species phylogeny with this dashed rectangle. And so this is the topology which we reconstructed from scratch in our reanalysis of their data. I'll briefly mention that this dashed green rectangle is not only interesting because it is the tree finch clade, it is actually the only clade where our reanalysis differed from the original published species phylogeny in the 2015 study. Otherwise, the topologies were exactly identical. In addition to the topology that I'm showing on the left, for here are reconstructed or re-estimated tree from their data, I'm also showing Bootstrap support values on all of the different branches. And in general, for the Bootstrap support analysis, you generally see pretty high support, more or less, throughout most of these, especially within this tree finch clade, as shown. Now, here is that same topology now with RAWR support values instead. And just take my word for it, if you kind of can remember what I showed you, take my word for it that basically the RAWR support and Bootstrap support values pretty much are very, very similar in all cases, except for 2 bipartitions within this tree finch clade. And so remember how I told you that this tree finch clade was the only one in which we had found differences, topological differences in our re-estimated tree versus what was originally reported in the 2015 study. Basically, this RAWR analysis is pointing us to the same estimate of phylogenetic reconstruction uncertainty. And it's basically telling us that some of this phylogenetic reconstruction uncertainty is due to alignment uncertainty. In conclusion, today what I've introduced to you is a new method referred to as RAWR, which consists of random walks for performing sequence-aware statistical resampling of input data. And we evaluated the use of RAWR for phylogenetic support estimation and assessed the performance thereof using simulated and empirical benchmarks. And so in our performance study, what we found is that RAWR offers better, comparable or typically better type 1 and type 2 error for its support estimates versus state-of-the-art methods. There was a little bit of a trade-off in terms of runtime. So there was increased runtime for RAWR analysis compared to the Bootstrap, but to some extent, this can be offset using reduced resampling replication. Finally, RAWR support pointed to the impact of sequence insertion and deletion processes on phylogenetic estimation uncertainty for a really charismatic clade of species, that of Darwin's finches. On the back of this study, we are continuing development of this method. And so some of the things that we're currently exploring are trying to add feature-aware / application-aware resampling to do better support estimation. Furthermore, we're trying to address the computational scalability bottleneck using parallel computing approaches. And finally, just going back to my note about how originally we had the design goal of having a very flexible general purpose resampling approach. With that idea in mind, we're continuing to explore the application of RAWR resampling to other sequence analysis tasks, not only within computational biology and bioinformatics, but in other areas as well. With that, I'd like to acknowledge the funding agencies that have supported this work, and I would be happy to take any questions that y'all have. I've also put up here a link to my website, as well as a GitLab repository where you'll find all of the data and scripts and software used to run this study. Thank you all very much.